Hello students, this is Faizan Mirza. We are discussing nucleic acids and protein synthesis. When we are discussing the topic of nucleic acids and protein synthesis, the first thing that we have to discuss is what nucleic acids are. So when we are discussing nucleic acids, DNA and RNA both are the type of nucleic acids that we'll discuss here, and we relate how they are related to the synthesis of protein which occurs in the ribosomes of either eukaryotic cell or a prokaryotic cell. We have already studied that previously. So DNA and RNA, they are the type of nucleic acids that we have to study in detail. This DNA and RNA, they both actually are chain of nucleotides. Several nucleotides join together, and when several nucleotides are joined together in a chain, they are joined by a bond, and the name of the bond is phosphodiester bond. So this bond join the nucleotides together. DNA and RNA, they both are the type of polynucleotides. When these polynucleotides are present in the cell, they basically are carrying the codes for protein synthesis, and they allow protein to be synthesized according to the codes that they have. Now, there are many nucleotides present in a certain polynucleotide. A single nucleotide is having three main components. They will be a phosphate group. We denote this with a circle in a P. This is having PO43 minus. It will also have a pentose sugar. If pentose means 5 carbon sugar, it is denoted by a pentose ring. This could either be a ribose sugar or a deoxyribose sugar. The difference in ribose and deoxyribose is on the second carbon. The second carbon of ribose is having the OH, and the second carbon of the deoxyribose is having hydrogen. This is the basic difference in the two types of pentose sugars which are present in the polynucleotides. And then there is a nitrogenous base or an organic base, and this can either have a single ring or a double ring structure. This is a single ring organic nitrogenous base. This is a double ring organic nitrogenous base. The double ring ones are called purine. The single ring one is called pyrimidine. The further types of these are thymine, uracil, and cytosine for pyrimidine, and guanine and adenine for purine. We abbreviate these, or we symbolize them with a capital letter, capital T, capital U, capital C, or capital A or capital G, respectively. So this is how a nucleotide look actually like. So it would have a pentose sugar, and this pentose sugar will have nitrogenous base attached on the first carbon, and it would have the phosphate group attached on the fifth carbon. So these are the symbols with which we'll be using in this chapter. We'll denote adenine with this symbol, guanine with this. You can see that they are of larger size comparative to the double ring structure, and these are the three you can say shapes for thymine, uracil, and cytosine. Just look closely at thymine and uracil have the same shape. We'll come to this later. This is how a nucleotide basically is. This is just repetition of the same. You can see that these are the bases. The purine bases are double ring, and the pyrimidine bases are single ring structures. This structure is of a polynucleotide. So single nucleotide which are present there. So DNA and RNA, what's common in them are the three nucleotides with they are common: guanine, cytosine, and adenine. These are the three ones which are common in DNA and RNA, whereas thymine is present in DNA only, and uracil is present in RNA only. These are the 5 basic nucleotides on the basis of whether they are present on DNA and RNA. What bonds are present in a polynucleotide? This is a single nucleotide. You can see here the bond that join two nucleotides together is a covalent bond called as the phosphodiester bond. When a polynucleotide is formed, it's like a string. It will have two ends. One of the end will be 5' end, which will have the fifth carbon of the pentose sugar, and the other end of the string will have the 3' end, the third carbon of the pentose sugar. The phosphodiester bond join the two nucleotides through the fifth carbon and the third carbon of the two nucleotides, and this is joined through the phosphate group. That's why it's called as a phosphodiester bond. This is the polynucleotide you can see here, and this string actually represents how this region is 5'. This is the end 5', and this here you can see that there's a 3' carbon, so we are representing it with 3' here. When we have moved on to the structure of nucleotides, let's just compare to a specialized structure of a molecule called ATP. ATP is adenosine triphosphate. It is the energy currency molecule. It is having 3 phosphate groups attached to a ribose sugar. Adenine is the nitrogenous base which is attached to the ribose at the first carbon. You can see this is the ribose sugar. This is the pentose sugar. The first carbon is having adenine attached. Adenine having 2 rings. On the fifth carbon, you have a phosphate group, another phosphate group, and another phosphate group. It's an adenosine triphosphate. So this is structure when adenine and ribose combine. This is adenine. When one phosphate is attached, it's adenosine monophosphate. When two phosphates attached to it, it's adenosine diphosphate. And when we have three phosphates attached, it's adenosine triphosphate. So ATP is made in respiration. We know that energy is stored in the phosphate bonds, and when these phosphate bonds are broken or hydrolyzed, the energy is released. A single phosphate bond, for example, if ATP is broken down to ADP, it would break down to ADP and it would release PI, and PI is an organic phosphate. When this inorganic phosphate is removed, 30.5 kilojoule of energy is released in ATP hydrolysis, and this is the energy which is used, which basically is used for vital process in the cell. You can just recall the structure of nucleotide that we have just discussed and compare its structure with ATP to understand, to develop more understanding over this. Differences between DNA and RNA are two types of polynucleotide. The first and foremost difference is the pentose sugar, being ribose and deoxyribose. These strands, double-stranded double-stranded DNA is always double-stranded, whereas RNA is single-stranded. But in some cases in viruses we have single-stranded DNA and a double-stranded RNA, but that will come later. So length of these polynucleotides, DNA is always long, RNA is always short. Nitrogenous base include ATGC in DNA and AUGC in RNA. Complementary base pairing exists. Complementary base pairing is when one purine base combined with another pyrimidine base. The structural restriction allows adenine to pair up with thymine or uracil and guanine to pair up with cytosine. 3 hydrogen bonds hold guanine cytosine together. 2 hydrogen bonds hold adenine thymine or adenine uracil pair together. When we are discussing hydrogen bonding is present in DNA between the complementary bases on the two strands, since it's a two-stranded molecule, whereas the complementary base pairing in RNA is on the same strand, since it's not having two strands. DNA, deoxyribonucleic acid. This is the molecule of inheritance which is having the genes, and these genes are having the codes for the synthesis of polypeptides that are produced in our body or in our cells. So the structure of DNA, it consists of two polynucleotide strands, and these two strands are twisted around each other. And when they are twisted around each other, so this is one strand, and you can see how the other strand coils around the first strand. You can picture these strands as having a ladder like structure. It's just that the ladder is twisted, and this is a double-stranded structure, and the steps of the ladder are the complementary base pairing that you have, and this is how DNA looks like. So DNA is a double-stranded molecule, and the two strands, they run in anti-parallel direction. Anti-parallel, I've already discussed that one end of one polynucleotide is 5 prime, the opposite end is 3 prime. So the twisted ladder, the two strand will be anti-parallel. One strand will end at 5 prime. Its opposite end will be 3 prime. The strand next to it will start at 5 prime here, and the 3 prime will be at the opposite end. So one is like one is like an arrow pointed upwards. The other is like an arrow pointed downwards. They both are twisted around each other to form a right-handed helix, and this is, for example, the two strands I'm drawing. This is the first strand of the DNA. Let's see, this is the white strand I'm drawing, and this is the yellow strand, the second strand, and this is how these are the complementary base pairing which hold the two DNA strands together. When the DNA molecule is pressed, these are the 3 prime and a 5 prime, you can say, ends of the structure. When complementary base pairing is present on the DNA, it actually is used to predict the sequence of the other strand. So if you know the sequence of one strand, you can predict the sequence of the other. Keep in mind, uracil can never be present in DNA. So if this is the structure of this sequence of bases of one DNA strand is given to you, you can predict the sequence of bases on the other strand. For example, if it's C here on the first strand, the opposite strand would always have a G here. If it's a G, so this should have a C. If it's a G, it should have A. It should have C. And if this is A, this should have T. So on and so forth. So you can just predict these, and you can come up with the outcomes. These are the hydrogen bonds which hold them together. The total number of hydrogen bonds between guanine and cytosine are always 3. The hydrogen bonds holding adenine and thymine together are always 2. This is the bond that hold the nucleotide together, the polynucleotide strand together here. This is the phosphodiester bond, as I've already discussed. This is a polynucleotide. You can see this is a single polynucleotide. This is the DNA molecule, a part of DNA molecule. One strand upright, the other and the other end, or you can say the other part is reverted. You can say it's upside down or inverted. This one is upright. The hydrogen bonds are present between guanine and cytosine and adenine and thymine. This region is called the sugar phosphate backbone. Sugar phosphate backbone simply means as it's having pentose sugar and the phosphate group. This makes up these hands of this of ladder. So this is how the DNA molecule is actually present, and this is a linear structure, and when it curls around in a right-handed helix, this is how it twists around. For example, if a DNA molecule was found to be having 100 nucleotides and 30 of these were thymine, how many guanine might be present there? You can just work out this, and you can see that there would be only 20 guanine molecules present there. DNA is a stable molecule. It undergoes replication, and when it undergoes replication, it produces a clone copy of its own kind. So this DNA molecule, this is a twisted structure. It would be a supercoiled structure at times. So the supercoil gets relaxed, and this structure start to unwind. When it starts to get unwind, the bonds between these complementary bases are broken by the enzyme helicase. It starts to just break these bonds, and the helix is released. These strands, they become separated. Free nucleotides of DNA which are roaming around in the nucleus, they would just come and they would start pairing up against these exposed ends. Against G, you would get a C. Against C, you get a G. Against T, you get an A, and so on and so forth. DNA polymerase will keep on joining these sugar phosphate backbone, while the hydrogen bond will hold the complementary base pairs together. And this is how the DNA keeps unwinding, and the molecule of DNA keeps growing. Eventually, you will have two DNA molecules made from one DNA molecule, and this is how DNA replication goes on. When you have DNA replication occurring, for example, if this one strand was red, this is the red strand, the original strand. The newly synthesized strand against both of these is like purple here, purple here. So when you have these strands here, so how do you think they would coil? One DNA would have one red and the other purple, and so on and so forth. So this kind of replication is called a semi-conservative replication. More of this in due time. You can see how this complementary base pairing occurs. A free nucleotide which is about to be placed in the replicating DNA, it must have two additional phosphates, and these two additional phosphates, they provide the energy, and with this energy they will come, it would just attach where it finds its place, and the DNA polymerase will join the sugar phosphate backbone, and the complementary base pairing will be held by the hydrogen bonds. Four enzymes are important here. Helicase releases the helix by breaking the hydrogen bond. Topoisomerase, it just releases the supercoiling. DNA polymerase join the sugar phosphate backbone. 5' to 3' carbons are joined together. DNA ligase fills up the gaps if there are any gaps present in DNA replication. So this is how your DNA polymerase, you can see it as it goes up. You can see one DNA molecule is being turned into two DNA molecules. You know this occurs in the S phase of the cell cycle. Complementary strands, I have already discussed that one strand sequence is used to predict the type of bases present on the other strand. You can just work these examples out. When we're discussing replication, we expect the replication could either be, this is the parent DNA, the new strand, the parent DNA would separate out. This is the white strand, the parent strand. The green one is the one which is synthesized new. So this green strand is paired against the white one. We can expect that it was initially expected that the white one was paired would just coil around each other. The green one would coil around each other. This would be the new strand, the new DNA molecule, the old DNA molecule. This was termed conservative replication, but it doesn't occur this way. It occurs in a semi-conservative way, in which you have this DNA molecule, it just separates out, and the DNA molecule that eventually gets made, both of them have half the original DNA and half of the molecule is made up of newly synthesized DNA. Here we have the dispersive replication. It was suggested that probably the replication could be dispersive, that some segments of DNA would have the original strand, some of them would have the old strand. But anyhow, it was proven with an experiment conducted by Measles and Stahl that the replication is actually semi-conservative replication. What they did in this experiment, they took bacteria and they allowed the bacteria to grow in nitrogen-15 medium. All the DNA was made for using nitrogen-15 isotope. When this bacteria was allowed to replicate for one generation using nitrogen-14 isotope, the new strand that got synthesized against the template strand. The template strand was nitrogen-15. The new strands got made were nitrogen-14. When these coiled together, so the coiled DNA was nitrogen-15 and 14 combined together. So this was hybrid DNA that was produced. So 100% hybrid DNA was produced in this first go. Then this bacteria were allowed to replicate once more in the same nitrogen-14 medium, and when they were allowed to replicate for one more generation, it was found that the original strands, they separate out this way. This is the nitrogen-15 of the original, this is the nitrogen-14, nitrogen-14 and nitrogen-15, which actually belong to these here. All the strands have separated. Now, since the bacteria is growing in nitrogen-14 isotope only, it means that all these four strands which have separated, they get the new strands against their template strands. which all be nitrogen 14 as represented by the red color here. So when they coil up together, so we get this kind of a structure of DNA that 50% of the DNA are nitrogen 14-14, 50% of the DNA are nitrogen 15-14, means 15% hybrid and 50% non-hybrid. You can just presume, or you can just process this further, and you can see how you can predict that. You can just predict the outcome of the replication if it is allowed to replicate for one more generation. So if initially it was 100% hybrid, then 50% hybrid, the next time it should be 75% hybrid. So here you had 0% of the nitrogen 14-14 DNA. Here it was 50%, so here it should be 75%. So you can just work it out in that way. This is the cesium chloride actual experiment of measles and Stahl. What they did, they allowed these bacteria, the bacterial DNA was separated using cesium chloride centrifugation. You can see that the band of DNA in the first generation was all nitrogen 15-15 DNA strand. They found a band here, and this is how nitrogen 15-14 it found a band a bit higher here. All hybrid DNA, these were 50% of nitrogen 14-14 and nitrogen 15-14 hybrid DNA, and this 75% 14-14 and 25% hybrid DNA. So this is how the semi-conservative replication was actually shown by the experiment of Michelson and Stahl. Next, we are discussing how DNA and protein synthesis go hand in hand. The bases of the DNA actually are present in the form of codons. Codons mean triplet of bases, and these triplet of bases, each triplet codes for a particular amino acid. So if your DNA is having this kind of triplets, the first is TAC, then CTG, then CA, then CTT. All of these will code for a certain amino acid. This dictionary is given to you. You can just use this dictionary, and you can see which amino acids are there. So the sequence of amino acids on a polypeptide are the primary structure, and the sequence of bases in the DNA are the ones that dictate the sequence of the amino acids in a polypeptide. How do you use this dictionary? You should know how to use this dictionary. You can just see that if you try to work this out, you can see how you have the first codon is TAC. On the first codon means there's a triplet of bases which code for a certain amino acid. So the first position is T, the second position is A, the third position is C. So in the first position column you look for T, this is T. In second position column you look for A, so this is A. So you go down in A, you go ahead in T, and you will look for this box. For the third position you have to look for C. In the third position box you look at C, and you come back here. So when you come back here, you see this is TAC. This amino acid is the one which is dictated by TAC, TAC. So this is met amino acid, which is exactly the same. So you can just work out all codons, and you can see which codons work for which amino acids. These are the total of 64 codons. These 64 codons code for 20 amino acids, and there are three stop codons. Three stop codons are ATC, ATT, and ACT. These stop codons they stop protein synthesis when protein synthesis has been completed. When we are discussing gene mutation, the next topic, gene mutation is just how the gene may change slightly and the nucleotide sequence may alter, and the altered nucleotide sequence will have a slightly different sequence because a single nucleotide change, and if a single nucleotide changes, which means that this gene is now have undergone point mutation. Point mutation, for example, GAG turns to GTG, for instance. Missense mutation occurs when the codon changes the amino acid as well. That's missense mutation. And silent mutation is when the codon has changed but the amino acid stays the same. Since some amino acids have a synonyms in that dictionary, so in that case that's silent mutation. Hemoglobin beta chain is having a normal allele and an abnormal allele. The normal allele is HbA, the abnormal allele is HbS. This is the dominant allele HbA, this is the recessive allele HbS. A person will always have two of these, like one of this and one of this, or probably both of these, or probably both of these. So if when a person is HbA, all the hemoglobin will be normal. If the person is HbS, then the person will be suffering from sickle cell anemia. If the person is HbA and HbS together, the person will be a carrier of the sickle cell trait. Now, what actually is happening here, you can see this is the codon sequence mentioned to you. You can see at this position of the codon is CTT. This CTT codon codes for the amino acid glue, but in the mutation occurs, CTT turns into CAT. You can see this is the point mutation that has occurred here, and because of this point mutation, this is not a silent mutation, it's a missense mutation. So instead of GLU amino acid, a val amino acid gets incorporated. GLU is a very polar amino acid, val is not a non-polar amino acid. So when a non-polar amino acid gets incorporated into a polypeptide, the primary structure changes, the secondary structure and the tertiary quaternary structure changes as well. So the folding of the protein is different. That's why the sickle cell anemia, the shape of RBC is different. Now, taking this discussion to protein synthesis, when we are discussing protein synthesis occurs, it basically comprises of two main steps, transcription and translation. Transcription is the synthesis of mRNA using a segment of coding strand of DNA, and when this is used, this means that mRNA transcript is ready. The transcript of mRNA goes to the ribosome where the polypeptide synthesis occurs, and amino acids are incorporated into a polypeptide chain. The process called as translation. So, for example, if this is the DNA molecule, this entire DNA molecule will have a certain region which will start to just uncoil. It would just separate from the strand, would just form a gap here, and while the remaining DNA strand stays coiled up. This is the coiled DNA, and this is the coiled DNA. Only a segment of this DNA got uncoiled because this segment is the gene. So this is the gene on the DNA that gets transcribed into an mRNA. So when the mRNA gets transcribed here, so the mRNA will be made using complementary base pairing, and this is the non-coding strand of this DNA of this gene, and this is the mRNA that got made against the template strand. Template strand is the coding strand of the gene which is used to make the transcript of mRNA. So this is the one that is having the codons. So codons are present on DNA, the coding strand, as well as on the mRNA. So this is the mRNA codons. These are the complementary codons which were made using the base pairing, the bases which are present on the template strand. So this is how the mRNA comes into the cytoplasm. What happens in translation? Once the mRNA is there in the cytoplasm, it is held by the ribosome. The ribosome hold two codons at once together, and when it holds two codons at once together, the two codons are then tRNA then comes. tRNA are the transfer RNA. Transfer RNA brings amino acid with them. They also have triplet of bases here. These triplet of bases on tRNA are called anticodon. Codons are present on the mRNA. So mRNA codon faces the complementary anticodon, and these anticodon on the tRNA cause certain amino acids to be joined together. Ribosome hold this assembly together long enough for a peptide bond to form between these amino acids. Once this peptide bond forms, ribosome will go and move to the next two codons, and then the next, and then the next, and then the next. This is how the polypeptide chain keeps growing, and the amino acids keep getting added to the chain. Eventually, a stop codon is reached where the protein synthesis will stop. Generally just speaking, transcription is how a part of DNA molecule is turned into an mRNA, and translation is how an mRNA is turned into a polypeptide. So you can see this transcription. This is the same process. This is the DNA, part of DNA uncoiled. The free nucleotides start to just form complementary base pairing here. The RNA polymerase enzyme here. The DNA polymerase works in replication. We studied for making the sugar phosphate backbone, but the sugar phosphate backbone of RNA will be made by RNA polymerase. So once this is made, you can see the mature mRNA. This is the mature mRNA which is made. This is the template strand, and this is the non-coding strand. You can see these labels, and you can just work it out yourself how the mRNA got made and transcription occurred here. So this is how once the DNA actually makes up the mRNA transcript, the mRNA just detaches. It moves out of the nucleopore and goes out into cytoplasm. We are talking about the eukaryotic cell here. If it was a prokaryotic cell, the DNA would already be lying in the cytoplasm. So this mRNA goes to the ribosome. The ribosome could either be attached to the rough ER, or it might be present free in the cytoplasm. In the cytoplasm, there are already tRNA. As I told you, tRNA are transfer RNA. They have, some of the tRNA may not have amino acids. The amino acids are also roaming about free in the cytoplasm. Amino acids get attached to their respective tRNA. So this is the tRNA which already is having the amino acid attached, and this is the anticodon which is present here. So there are, as we know that there are 64 codons, so there are 64 anticodons as well. So for every anticodon there is a tRNA having it, and the amino acid is present there as well. So this is how the mRNA is held there. You can see that the first two codons are held. The first codon is AUG. So when AUG is exposed on the mRNA, the tRNA, which will have complementary base pairing of UAC, will come here, and it will bring the first amino acid MET here. When MET is present here, the second codon is also exposed. The second codon on mRNA is UAC. So the second tRNA that comes will be AUG. It will have the anticodon AUG. It brings the other amino acid TYR. This assembly is held by ribosome long enough for a peptide bond to be formed. The enzyme peptidyl transferase is used for making this peptide bond. Once this is made, we have the ribosome move to the next codon. The next codon is AAA, and you can see that the tRNA that was attached to MET goes out of the ribosome, and the second tRNA is still in the ribosome, and it's holding this polypeptide which is still growing. So this tRNA comes. This tRNA will have UUU here, since the anticodon will be UU, since the codon is AAA. So the complementary base pairing will occur. The amino acid will join together. The polypeptide chain will keep growing. Here you can see that UCA is having HEU, and then AGG is having UCC, and then eventually you will have the stop codon, which is UAA. The stop codon will be a signal for the ribosome to leave the mRNA. So mRNA will get detached, the polypeptide will get detached, and this polypeptide will then be transferred to the Golgi for further processing and modification if it's a eukaryotic cell. Ribosomes, they work alone if they are free in the cytoplasm, but if they are attached to the rough yard, they work as polysomes. Polysomes just mean many ribosomes working together, and if they are working together, it means that a single mRNA will be turned into several copies of the same polypeptide. Here in this diagram again you can see the same thing. This diagram, although is a bit of messy, that they have shown more than 2 codons being held in the ribosome. So anyhow, the basic concept should go that if the DNA was having the codon TAC, the mRNA would have the codon AUG complementary to the DNA. This is the template strand of the DNA. So this is AUG. When AUG comes here, the tRNA that comes here will have UAC. So UAC is exactly, this is the tRNA, this is its complement. Anticodon is complementary to the codon. So this UAC is actually almost same as the template strand TAC. The difference is that the A is there, the C is there, the A is there, the C is there. But since DNA, it was DNA, so this was thymine. Since it's tRNA, it's so uracil. The RNA, they cannot have thymine, and the DNA cannot have uracil. So this is how the protein synthesis will go on. RNAs of several types, and they are all single-stranded. Mostly they are short structures. In our parts of the syllabus, we just have to study three type of RNA: mRNA, tRNA, and rRNA. mRNA is the transcript of segment of DNA that strand with a gene. This is messenger RNA. tRNA is also a transcript, but it basically brings the amino acid to the polypeptide chain. Each attached amino acid to the 5'. The amino acids are attached to the 5' end of the tRNA, and rRNA are the ribosomal RNA that makes the ribosomes. We know that ribosomes are present in the nucleus. The RNA is present in the nucleus, is present in the cytoplasm. It might be present in ribosome as well. So this is the location of RNA, whereas DNA is present in the nucleus. Messenger RNA is a linear structure. This will have 5' end and a 3' end. This single strand molecule will be a straight linear molecule, but it will coil up. When it coils up, it will coil up in this structure. Why it coils up? Because of the hydrogen bonds that form between the complementary base pairing present on the same strand. The same strand will fold up because of the complementary base pairing, unlike DNA, where the complementary base pairing occurs between the complementary strands. Just the way we have the codon dictionary of DNA, we have the codon dictionary of mRNA as well. The first position, the second position, the third position. The mRNA codons are exactly the tripletal bases of complementary codons to the DNA, of which is the coding strand. So if we have TAC, GAG, this sequence was given to you, for example, on your template strand of the DNA. We know that the RNA that would be made will be having this AUG, and the bond will be made by these red bonds. These are the ones which are made by the RNA polymerase. The mRNA will grow in this way, and once this mRNA have been made, this mRNA will be transported into the cytoplasm, where it will be translated into polypeptides. So if it's AUG, CUC, GAG, UUU, and UCC, and UAG. So these are the amino acids that get incorporated. Since this is the mRNA we are using for the amino acids, so we'll use the mRNA dictionary. Previously we used the DNA dictionary, and that DNA dictionary, the appendix, was the one for the codons of the DNA. This is again the repetition of the same, how the ribosome is continuing the process of translation. These are the two questions that you can just practice if you want to develop a better understanding of this, the basic concepts of. This is how a tRNA is present. tRNA is also made from the genes of the DNA only. It's also a transcript initially. So when when that segment of DNA makes a tRNA, the tRNA comes out in the cytoplasm. It folds up into a hairpin loop structure or a clover leaf shaped structure. This is the 5' end of the tRNA. This is the 3' end of the tRNA. It rolls up like this. Although we draw it in this way, the simplified diagram, but actually it's like this. You can see the complementary base pairing. This is CCA. So it will have GGU on the other part. These are not two strands. Keep that in mind. They look like a two strand. They are a single strand. This is the sugar phosphate backbone. So the single strand molecule having complementary bases on the same strands, and this is why the molecule ends up folding. This region of the tRNA, this is the region which will always have the basis CCA, and this is the site where the amino acid gets attached. So this is a specific binding site of amino acid. This is the region where you have triplet of bases, which are the anticodon, and these will be complementary to codons of the messenger RNA. That pretty much is it. Thank you so much.